Hi everyone, I'm Charlotte and welcome back to Situ, Operation Wallacea's online lecture series. This week I'm introducing Dr. Burton Lim. Burton works out on our project in Guyana, which is one of the most remote projects that we run deep in the Amazon rainforest. Burton is going to tell you about his research out there and the rest of his work at the museum. So please enjoy the lecture and come back and watch the rest of our series.
Hi, thank you for tuning in to my presentation as part of the OpWall In Situ program. I'll be talking to you about the work that I do at the museum. So my name is Burton Lim. I'm Assistant Curator of Mammalogy at the Royal Ontario Museum in Toronto, Canada. So I'll start off by just giving a bit of a sort of a background on where I work. So the Royal Ontario Museum, we usually affectionately refer to it by its acronym, ROM, or the ROM. So we're Canada's largest museum that combines both natural history and world cultures. So a lot of times you'll see in cities, usually those types of museums are separated into two different buildings. But the ROM is a little bit different in that we actually combine everything under one roof. So we have about 12 million artifacts or specimens in our collection. There's an international sort of focus. So we have projects in about 27 different countries throughout the world. There are 32 curators that specialize in art, archaeology, and science. So I'll just be talking about my position, specifically about mammals. But all the other curators, they'll be doing, something, quite different from what I'll be describing as my job as a curator. So my background is I got a Bachelor of Science degree at the University of Toronto, but I did things a little bit differently. I didn't go immediately into a graduate program, so I actually got the job at the ROM first, and I was fortunate that I was able to go back, for graduate studies while still working at the ROM. And another thing I sort of did a little bit differently is that I also enrolled in this Fellowship in Global Journalism program. So during my presentation, I'll be talking about not only some of my own research, like in peer-reviewed scientific journals, but I'll also, whenever possible, also talk about examples of when I've published, some of my research in like either newspaper articles or magazines. So one of the things I've always had an interest in was sort of the science communication aspect of the work that I do at the ROM. So whenever possible, I always try to sort of present my research to a wider general audience, beyond academia. Okay, so as a mammal curator, I've listed down 7 sort of topics or categories of the work that I do. And what you'll notice is that a lot of these topics or categories are all sort of intertwined together. So, for example, I'll be talking about field work, but a lot of the field work, leads to the research that I do. But a lot of times it'll also contribute specimens to the collections. So a lot of these categories, they are standalone, jobs or tasks that I have, but a lot of it is all intertwined and all mixed up into what I do as a mammal curator. So I'll start off with collection management. So we have about 125,000 mammal specimens. So the ROM is over 100 years old. Over the years, we've accumulated things primarily in Ontario, the province, but also in Canada. But now, more recently, we've been, working throughout the world, looking at the biodiversity of mammals. Most of the stuff I work on are the small species, such as bats, but also rodents and shrews and opossums. But as I'll tell you a little bit later, more recently, I've also been working on blue whales or whales in general. But I'll get to that a little bit later. So one of the things I want to highlight in this photograph is in the upper left-hand corner here, we have a species of bat called the little brown bat. So about 10 years ago, this was the most common species, found primarily in North America. but there's been this disease called white nose syndrome that has been decimating the populations of hibernating bats. So back in 2014, 3 species of bats were put on the endangered species list in Canada because of this white nose fungus. So I bring that up just to highlight the fact that one of the sort of important aspects of museum collection is that it captures sort of the historical record of where species are found and when they're found. And for white nose syndrome, if we want to find out, the impact, across the country, we will be able to go back into museum collections to find out, okay, well, these species that are endangered now, or they were found in these areas. Are they still found in these areas? Are they still abundant? So, if these specimens hadn't been collected, 100 years ago or whenever, we wouldn't be able to say for sure, where some of these species were found and when they were found. So it's a historical record that if you don't collect that information at that time, that information basically will be lost. You can't obviously go back in time. So that's one of the sort of strengths of the museum, is that it represents an historical collection of species diversity. And this was an article that I had written for the Toronto Star newspaper. So again, this was when people were trying to figure out what the impact of all these bats that had died. So it was estimated that millions of bats had died in North America. So I just wrote this story about sort of the economic impact of these bats, and this was published in the newspaper article that you see in front of you. So in addition to the actual specimens, about 30 years ago, we started collecting tissue samples, also frozen tissue samples. So we've also gotten into the area of looking at DNA study. So we found that in general, the number of mammal species has probably been underestimated by at least 25%. Some people, think that there are 50% more mammal species that are known. So that's quite amazing, being that mammals is one of the the better studied groups of animals around, but we still don't know like 25 or maybe even 50% of it. So a lot of this undiscovered species diversity has been because of sort of genetic work looking at DNA samples from frozen tissues. And on the right-hand side, you can see that there's all this smoke. So that's liquid nitrogen, so it freezes things at like minus 196 degrees Celsius. So basically, when you drop something in there, that tissue sample will instantaneously freeze. So that's the best way to preserve DNA, is to quick freeze it at as low a temperature as possible. So not only do we have the voucher specimens, but we also have tissue samples for a lot of the specimens that we collect, especially over the last 30 years. Some of the public programming stuff we do. So again, everything that I'll be talking to you about today, all of it is based on specimens one way or another. So the City of Toronto had this biodiversity publication series. So these were just booklets that were available for free to the general public. So you can see there's a whole series of different booklets that were on not only mammals, but there's trees and shrubs, spiders, etc. So I helped work on the Mammals of Toronto booklet. So again, we went back into the historical records that were based at the museum to look at what species were found, where were they found. So that information was part of this Mammals of Toronto booklet series. Some other public programming things that I've done. We've had this program called Ontario Bio Blitz. So basically, a group of experts are assembled. So again, not just for mammals, but birds, plants, insects, and over a 24-hour period, we try to document as many species as possible to get an idea of the diversity in a particular area. So in Toronto, we concentrated on the different watersheds that pass through the Toronto area. And yeah, so this was just an example from 2017. Then some photographs of some of the bat work we do. So there's different levels of participation from the public. There were some students that would help us out. You know, they would be helping us, you know, net bats at night, trap rodents in the daytime. But there was also more public awareness programs where we had guided like bat walks at night. So that was more of a sort of a citizen science or public awareness type program, just to get the general public interested and for them to know, you know, the type of work that we do at the museum. And of course, yeah, so a lot of those public program stuff, that's, you know, more like, you know, community outreach programs. But of course, you know, within the museum building itself, we have our own galleries and exhibitions. So one of the main ones is the Shad Gallery of Biodiversity. And you can see sort of the main entrance to this. So we started off with the white rhino. So this was an animal that died at the Toronto Zoo. So we were able to acquire that to put on display for this gallery, which opened up in 2009. So we were able to talk about a conservation story, and this was actually a sort of a successful conservation program where the white rhino, the populations got down to below 100 in Africa, but I think now they're in the tens of thousands. So I think that's one of the success stories for conservation, was the white rhino. But then we talk about, you know, different ecosystems in this gallery as well. The ROM has, I think, one of the few bat caves that I know of in museums. So this bat cave is actually based on a real cave in Jamaica, St. Clair Cave. So the field work for this cave was originally done in the mid-1980s. And we renovated the bat cave at the ROM in 2010. So I was able to go back there and do some of the field work to get video footage and other information for the upgraded bat cave at the museum. And yeah, so the bat cave we have is actually based on real parts of the St. Clair Cave. So this cave in Jamaica is about 3 kilometers long. But of course, you know, we only recreated, I think, about half a dozen different sort of sections of the real cave. So here you can see this real stalagmite in the upper middle photograph. So that corresponds to what we call our cavern in the bat cave at the ROM. So you can see, you know, over the 35 years since we did the initial field work for it, it looks like the stalagmite has changed a little bit from what it looks like back in 2010. But again, the artists that went on that trip, you know, they took molds, you know, to try to, you know, recreate the authentic, you know, feel of the cave in Jamaica, St. Clair Cave. Yeah, so the bat cave is one of the more popular exhibits, displays at the ROM. So I think behind dinosaurs, I think it's the second most popular one at the museum. In addition to like our permanent galleries, such as the Bat Cave and the Shad Gallery of Biodiversity, we also do temporary exhibitions. So this one, as I mentioned, I've worked on whales. So we had a exhibition on blue whales in 2017. So this was actually based on whales, 2 blue whales that actually washed ashore in 2014 on the west coast of Newfoundland. So I've got this short video clip that hopefully we can play here. Yeah, so this just gives you an idea of, yeah, so this was beginning of May in 2014. The night before we arrived, it snowed. As you can see, the whale carcass is still half in the water, and it's in front of this, very rocky shoreline and this boardwalk. So when we got there, we knew that, we couldn't, salvage the bones, in the current situation that it was left in. So we were able to get the carcass towed by the boat you can see in the lower left-hand corner. So it was about a 2-hour boat ride into the open water. So we were able to get it into this more protected inlet, and we were able to drag it up onto this this closed fish processing plant, the boat ramp. So we were able to work on this carcass. So we had 10 of us working 6 days to get this 100-ton blue whale carcass down to the bones. So these are just some of the few gruesome shots of the process. But sort of amazingly is that a lot of the techniques we used were based on sort of the the whaling techniques that were used like over 100 years ago. So just getting these big flensing knives and cutting up sections to peel off the skin and the blubber to get down to the flesh, and then we just skinned the meat down to the bones. So one of the only main differences that we had heavy equipment like this front end loader that helped us pick up things like this big jaw coming out of the head of the blue whale. And I was able to write an article for the National Post newspaper in Canada. So for those of you that want to learn more about that whale salvage operation, you can find this on their website. So I was able to give a first-hand personal experience or documentation of salvaging a 100-ton blue whale. And of course, so we salvaged the bones in 2014. So we put it on display in 2017. So here you can see it here. We also salvaged the heart of the second blue whale, and that was plastinated in Germany. So some of you might have seen Body Worlds. So basically, it was the guy in Germany that had invented the plastination process of preserving organic material. So we have the blue whale heart, which obviously is the largest heart of any animal that has ever lived, and the blue whale is the heaviest species of any species that's ever lived, so heavier than any dinosaur. So we had this exhibition on in 2017 looking at basically the biology of the blue whale, looking at the evolution, the conservation of blue whales, communication, its feeding behavior. So that was 2017. And in fact, if anybody is visiting Toronto in 2021 next year, we are actually in the process of planning a new whale exhibition. So since we got the blue whale skeleton, we've gotten a right whale skeleton that was killed during a ship strike in the Gulf of St. Lawrence. And then we also salvaged a sperm whale skeleton. So now we'll be able to talk about whales in general, about their biology and their evolution. So that will be opening up next year, hopefully around June. Okay, so let's get into some of the field work that I do. The upper right-hand map shows, so the green countries are the countries that I've done field work in. So I think I'm getting close to about 30 different countries, throughout the world, but most of it has been in the Americas, primarily South America. I've only had one trip to Africa. But I've also done quite a bit of work in Southeast Asia. But what I'm going to be talking about here is about a trip in 2015 to Sri Lanka. I'll be talking about this one in particular because I had a couple of graduate students from the Environmental Visual Communication program. So that's a program that is out of Fleming College in Ontario, but it's based at the ROM. So we were able to get a couple of the graduates from the year before to come on the trip to document the expedition, primarily from a social media perspective, but also just from a sort of a science communication perspective. So that trip was funded primarily by the National Geographic Society, but we also got funding from the ROM Governors, which is the sort of the fundraising arm of the museum. Okay, so the central maps just sort of shows you the places we went to. So it was like a 1 month trip. So we covered quite a bit of the island. We were just really missing sort of the northeastern part of it. But we covered, the Central Highlands, and then it, slopes down to the coast, obviously. But there was a few dry areas in the southeast that we went to. And then there's a few photographs of some of the things we did. So we were taking wing punches for DNA study. The next two slides, I'll be talking about the two main methods that we use to catch bats. So these are mist nets. So these are the same sort of mesh type nets that are used to catch birds. So what I have here is this is what we call a triple high net system. So basically, it allows you to stack 3 regular nets on top of each other. So you can get up to about 9 meters off the ground. So you can get some of the more high flying bats. So that just increases your chances of getting more bats, but also different kinds of bats that are flying higher up off the ground. As you can see, it's this spot in Sri Lanka was quite windy, so it was this open area. You can see, so the net is actually blowing quite a bit. So even though it's a very fine mesh net, it still catches the wind quite a bit. So you can see that it's actually billowing out quite a bit. And this is a quick time-lapse video that the EVC students took just to show you how long it takes to put up a triple high net. Yeah, so normally this takes about half an hour, depending on the situation. So the toughest part is just getting the pole set up and stabilized at the beginning, and then you just string the nets across. And this is a Harp Trap, so it gets its name because basically it's fishing line that is strung from the upper metal rods down to the bottom, and usually these are used where there's a high concentration of bats. So this is a cave entrance in Sri Lanka. So when the bats come out, if their echolocation doesn't pick up the fishing line, they'll hit the fishing line and that'll stop their forward momentum, and they'll fall into the green bag below. And you just easily go in there and grab the bat out of the bag. So it's a little bit easier to maintain, to manage during the night, compared to a mist net where you have to actually untangle the bat out of the net. So the Harp Trap is sort of easier to work with in situations where you get a lot of bats coming in, such as a cave in this situation. And this is another time lapse that the students put together on how to put this up. So yeah, so this one is a little bit easier. So I can usually get this up within about 10 minutes or so. Okay, and then I'm going to end off on the Sri Lanka field trip by showing you another 1-minute video that the EVC students put together just to sort of highlight the expedition in 2015. Okay, so I don't think this video is working here. Okay, so we'll just move on. Okay, so you can see the video on YouTube. So the ROM has a YouTube channel. So you can watch it here, but the EVC students also put on a lot of material like on Twitter. So you can go back if you just search on the hashtag ROM Sri Lanka, you'll be able to pull up all of the information from that field trip in 2015, but also the ROM website on the blog. There are several stories that documented our 1 month trip there. Okay, so now let's get into some of the teaching that I do. So the teaching I'll be talking to you about, basically there are field courses, so it's all hands-on stuff. Yeah, so this is a field course that I've been helping teach back, that started in 2012. So this course we have every other year. This year we actually got lucky because the year before we decided to postpone it a year, before the COVID situation happened, because we couldn't align all of the instructors' schedules together. So we thought we'd better if we just delayed it a year. So it actually worked out quite well. Is that we missed all of the pandemic situation. But basically we've run it 4 times. So again, a lot of hands-on stuff. So the students, and most of the students are from Latin American universities, but we do have students, from North America, from Europe, that also attend. But they get to do all kinds of different bat field methodologies, such as doing echolocation call recordings, preparing specimens. So again, a lot of hands-on experience and training on this field course. Another course that I taught, so this was a course that was back in 2017. So this was a course that was done at the University of Guyana. So it was primarily for their students, but there were also people that were involved in the National Protected Area System, like some of their forest rangers. So they got to learn mammal survey techniques from not only myself, but also Matt Hallett at the University of Florida. Yeah, so that was a 2-week course. So that started off with lectures at the University of Guyana campus in Georgetown, but then we spent a week at a field station about an hour outside of the city, south of the city. And I think there was about maybe, looks like there was about maybe 20 students that were a part of that. And the third example for teaching is that at the University of Suriname, so that's the country right beside Guyana in northern South America. So they started a Master in Biology program back in 2012. So I've taught the mammal elective course a couple of times. Yeah, so this is just some photographs. So this is just not bats, but we also do small mammal trapping for opossums and rats. But we also set up camera traps for large mammals. So that's at the University of Suriname. And yeah, so I don't have any students of my own, but I have been on graduate committees for other students at other universities. So I've just put up a few examples here. So at the University of Toronto, so I've been on the graduate committee of a couple of students there, and of course the University of Suriname, so a student that was involved with that mammal elective course, but also students from Brazil, the US, and also Mexico. I've served on their graduate committee. So if there's any students out there that are watching and you need an external mammal advisor, you can look me up. Okay, so then I'll sort of finish off, not sure how we're doing on time, but I'll finish off with research for the last, hopefully about the third of the lecture here. So most of my research is looking at evolutionary relationships, looking at the phylogenetic relationships or the phylogenies of how mammals or bats in this particular case are related to each other. So this is actually part of my PhD dissertation. So I looked at a group of New World tropical sheath-tailed bats. It was based on a molecular phylogeny, which you see on the right. But I used that phylogeny to look at the evolution of different character systems. So this looks at the foraging behavior. So for this group, I found that these sheath-tailed bats in the New World tropics, they broke into two main sub-tribes. So one of them, and this corresponded to the species that forage in open areas, so usually above the the tree canopy or in open areas. But then there was another sub-tribe that were the species that foraged within the forest, so within cluttered areas. So that was like a direct link between either open or cluttered foragers. But when you also look at their echolocation call parameters, you'll see that, again, there's that correlation. So one sub-tribe had higher echolocation frequencies, they had shorter call durations, and they had shorter pulse intervals, and that's compared to the other sub-tribe. So basically what this says is that both the foraging behavior and the echolocation call parameters, they had a phylogenetic or evolutionary history to them, so it wasn't as if each species individually, had ecological adaptation. So a lot of these foraging behaviors and echolocation call parameters, they have an evolutionary perspective to them. So basically, the common ancestors of these two sub-tribes, they go back, more than 10 million years, but they're the ones that were either, the open or the cluttered foragers with these echolocation call parameters. So a lot of the ecological characteristics have an evolutionary history to them. And another thing I sort of do research on is another area of evolution called biogeography. So again, just basically looks at where are species found, and basically why are they found there. So what caused their speciation or their evolution. So this looks at a group of mastiff or free-tailed bats in the Caribbean. So you can see I've marked off the different islands. Martinique in red, blue is Jamaica, and green is Dominican Republic. So these three colored lineages or taxa, they were considered the same species, but as you can see that genetically they're not even each other's closest relative. So basically, superficially they look similar, but based on their DNA, they're actually quite different. So that just looks at the biogeographic island origin in the Caribbean. So their speciation was actually driven by isolation on these islands. Okay, so another thing, so the two examples I just gave you, we're looking at sort of the evolution and their phylogeny, but sometimes some of the work that I do is more just looking at sort of the species diversity or the biodiversity. Some of the stuff is describing new species. So some of these phylogenetic studies, I'm looking at the relationships, but I realize that there's no name attached, to some of these taxa or lineages. So that involves having to actually give a new name for a species. So this is a project that I worked on with several co-authors. So this was so obviously these aren't bats; these are mouse opossums, but these are from the highland region in Guyana. That is also part of the highland region in Venezuela. So this is a fairly remote area that's difficult to get to, so very few people have studied there. So I was there about 15 years ago, and some of the new discoveries we got, we actually described a new species of mouse opossum, so the one on the left here. And this is the paper that formally described it in the scientific literature. But I was also able to write a newspaper article for the Globe and Mail in Canada, talking about how I went about describing a new species of mouse opossum. So again, this is available online if you want to read up on it, as is the original article describing the new species. And one of the tools I sort of mentioned that I look at genetic data. So one of the techniques I use is something called DNA barcoding. So this sort of method or approach was developed to identify, just about any animal species. So based on a very short 657 base pair fragment of mitochondrial DNA. So it works, most of the time. So I think it's probably effective about 95% of the time. So that's actually pretty good. So when it doesn't work, then you know that you need to look at something else, some other molecular data set, or you need to look at morphology or something else to see why is this group different? How come it doesn't fit with the DNA barcoding identification system? Yeah, so this looks at the species diversity of tropical bats based on DNA barcoding. Yeah, so this is a paper that came out in 2011. And I've also written actually several newspaper articles on DNA barcoding. So this particular one was published in the Dallas Morning News. And yeah, so this was just talking about sort of the practical applications of DNA barcoding. So beyond the use of for scientific identification. So this just sort of documented one of the practical uses of being able to identify seafood or fish, and studies have shown that a lot of the sushi fish is actually not top grade sushi fish. There's a lot of other substitutes that are going on. So DNA barcoding has actually identified this. So it's actually being used as a tool to identify things that might be difficult to identify, especially if it's just like a part of a fish or an animal like the fillet, that you don't have the whole animal, that it's very hard to identify from just a piece, like a chunk of meat or something. Okay, and then as far as biodiversity sort of research goes, another thing I look at is the community ecology. So not necessarily the evolutionary relationships, but looking at how many species are found in an area, and what's the relative abundance of those species, and how do those areas compare based on either bat species diversity or relative abundance, or other small mammals like rats and opossums. So this was a study that was done as a larger project with the World Wildlife Fund. So I think this project was actually in 2013, I think. So there's a bit of a lag to get this report published in 2016. But again, it was like a larger biodiversity assessment. So other people were looking at birds, insects, plants. So one of the things that the WWF wanted to look at was are there areas that needed more protection as say a national park. So this graph is just a very quick representation of two of the sites we worked. So you can see that the top line that I've marked off in red here is Parabara. So the species accumulation curve, number of individuals by species, it seems to be higher than Cusad. But if you look at other measures such as species richness and diversity indices, it also indicates that Parabara does have the most species and the highest species indices. So say to say that it probably is different and higher than found at the other place called Kusad. And that field trip, I was able to publish a story about not only the work that I was doing, but the work that the other people were doing on part of the trip. So that was a story that appeared in the Caribbean Beats in-flight magazine. So just another sort of outlet or venue to sort of get the science communication out there for a more general audience to read. Okay, so that brings me down to the Operation Wallacea Guyana project. So that is my connection. So the Guyana project started in 2011. So I've been fortunate that I've been able to work every year of the project. So we've done 9 years. So next year will be the 10th year of the project. But again, it's a larger biodiversity monitoring project with, again, people working on birds, insects. There's a forestry plot study going on. So basically, we're forming, a good solid baseline database of the health of the Iwak-Rama forest. So this information will be good for if there are any changes to the environment, say either by, local disturbances, such as maybe mining operations or, toxins that are in the river system, such as mercury, or if there is any, logging going on, or more generally, if there's any climate change, we will already have like a 10-year baseline study as a comparison. So a lot of times that's the problem, is that when people want to find out whether this is a local disturbance or a sort of a climate change issue, is that they don't have like the baseline data. So I think that's one of the important things about the Opwal Guyana project, is that we've built up a 10-year sort of baseline database of the health of the ecosystem, the biodiversity in Iwak-Rama forest. Okay, so Iwak-Rama forest, the country of Guyana is located in northern South America, right here. And Iwak-Rama forest is pretty much smack dab in the middle of the country. And then within Iwak-Rama forest, we have 6 study sites. 5 of them are within the Iwak-Rama forest proper. There's one site, so it's actually in collaboration with the Iwak-Rama and the Suriname village. So it's one of the indigenous villages in the area, but just outside of the Iwak-Rama forest. So these are the 6 sites that we've worked. So what we've done is we've standardized the survey methodology. So basically the bat netting grid is 100 meters by 100 meters, and every 50-meter station we have a couple of nets. At each of the 6 sites, usually we work about 5 nights. So we try to just do everything the same way to standardize the data to make things comparable from year to year, from site to site. Okay, so just quickly summarizing this. So in the 9 years we've done the study, so we've caught almost 3,000 bats, representing 54 different species. And every year we've added like a new species. Like even last year, the 9th year, we still added species to our list. So we're up to 54 so far. And this looks at the relative abundance, and again, this sort of skewed distribution is typical for not only bats, but most other mammals, most other organisms, birds, insects, plants. Is that usually you find that you get only a few species that are very abundant, but you get a lot of species that are considered very rare or just hard to catch. So this skewed distribution is typical of most organisms in communities. So the picture of this bat, it's the most common species that's been caught. So so it represents about 20% of the captures of bats as one species. But I've marked off this red box. So the four most common species, they're all fruit-eating bats. So again, in the understory netting grid that we use, the fruit-eating bats are the dominant species. Okay, so this just gives you an overview of the year-to-year data. So just what I want to highlight is that I boxed off the year 2012 and the year 2018. So 2012 had a low number of species, so only 25, and a low number of individuals, so almost 200. But in 2012, it had the most species and the most individuals. So you can see that between these two extremes, so 2018 had twice as many individuals and 50% more species diversity. So that just gives you an idea that I was a little bit surprised as we were collecting the data over the years, is that that there is quite a bit of just natural variation that goes on. So just imagine if you had come 2 years and say these are two switched around. 1 year 2018, you had all this stuff, and then the next year, you had half as much. You'd be wondering what's going on. But the longer term data that you have, you realize, oh well, everything, that's just normal fluctuation. So that's I would say nothing to be concerned about. So that's just sort of typical. And if you look at 2017, so the purple line here wasn't that much better. So I had a few more species than 2012, but it actually had fewer individuals caught. But that was like back-to-back years. So 2017 was one of the worst successful netting years, but then the next year right after that, it bounced back to be the best year. So again, just an indication of there's a lot of variation in the natural background, so the species diversity and relative abundance for bats. Okay, so this looks at the different sites that we worked. So the last 2 years, Suriname Lake and Suriname Village, we only did those for 1 year each. So we ended up settling on these 6 sites as the main survey sites. But what you can see here is that even among sites, there's quite a bit of variation. So Rock Landing had almost twice as many individual bats caught than any of the other sites. But the species diversity, so 37 species, is still the same as in Turtle Mountain, even though we caught about half as many bats at Turtle Mountain. The species diversity was the same. So again, just interesting comparisons between the different sites. But you can see Mill Site is where they do the reduced impact logging. So that seems like a natural trend, that it has lower species diversity and lower number of bats compared to the other sites. So again, just to get an indication of some of the among site variation that's going on during this study. Okay, and then I guess the last pattern of interest I want to show you. So this is just looking at Turtle Mountain. So again, I've boxed off 3 years of an interest. So there seems to be a 3-year cycle: 2012, 15, and 18. So again, remember Archibius planerosus, that was the fruit-eating bat that is the most common during the study. But in those 3 years, we didn't catch any of them. But that's also true for the other 2 species of large Archibius, which are fig-eating specialists. Again, those 3 years, we didn't catch any. And then, so Archibius obscurus, so we caught one of those in 2018, so that sort of, spoiled sort of the trend. But again, you can see that, 1 year they disappear, but then the next year, like in 2019, Artibius planoropsis, exploded. So we got like 68 individuals, during the 5 nights of survey. So again, if you only come like, 1 year, 2018, and say, well, what happened to all the big fig eating specialists? You would think that something was going on, but no, it's just that there's this 3-year cycle that we picked up, with this long-term data. And then, obviously the next year, they came back, full force here. So again, so it shows the value of getting long-term data is that you get to see these trends, and then you can also try to explain some of these trends. So I think here, I think we need to do some phenology studies looking at what's fruiting in the area. Yeah, so I have a feeling that where we have a bat grid net set up at Turtle Mountain, we're sort of on the edge of where the fig trees are probably going through a 3-year masting cycle. So basically, if those fig trees aren't fruiting, these big species of Artibius, they just fly somewhere else. So they're a little bit different from birds who are usually more localized. Some of these bats might fly several kilometers a night just to go to a fig tree, but of course they'll come back the next year when things are fruiting again. So again, with the long-term data, if you see certain trends, it'll enable you to better explain the data and why some of these trends occur. So again, it makes long-term studies much more powerful as far as looking at the data. The Opwall field trip I've documented in the ROM magazine. So again, just looking at not just the bat study, but looking at some of the other studies. So this is actually the bird team. So we actually caught an owl in her bat nets. So they're identifying this owl that was caught in their nets. So again, I tried to do some science communication by publishing in magazine articles for a wider general audience to read. But we've also done some research from these Opwall Guyana projects. So the first author on this paper, Tom, so he actually came on the first trip in 2011. So he came as a student volunteer, but then for the next 3 years, he came as my bat assistant. And during that time, he worked on his master's thesis, not using the Opwall monitoring project, but he actually looked at sort of the seed dispersal, so sort of a secondary project as we were collecting the Opwall data. But he was looking at seed dispersal. So I found out that one of the more common seeds that was found in the poop of these bats was a species of Cecropia. So this is what the seed looks like that it poops out. And this is the Cecropia tree. So this is a tree that usually flourishes in open areas. So Cecropia trees are important, as a colonizing pioneer species. So these pioneer species need to take hold in sort of the open areas to create the shade, for the successional trees, the taller emergent trees to take hold. So this is all part of natural forest succession. So this species of Artibius littoratus, is an important seed disperser for this pioneering tree species.